Good evening, and welcome to Dreamy Academy. Tonight we turn our gaze inward, exploring the quiet marvels that shape our every breath, our every gesture, the very architecture of our being. As we begin, I'd love to know where you're watching from. Your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Consider for a moment the simple act of lifting your hand, a gentle rise, a turning of the wrist, the delicate curl of fingers. It feels effortless, almost thoughtless, a natural extension of your will. Yet beneath the surface of your skin, an astonishing universe of precision and power is at work. Every flex, every extension, every subtle shift of balance is orchestrated by an ancient, intricate system. Your muscles, they are the silent architects of motion, the tireless engines of life, constantly shaping your interaction with the world, even as you lie perfectly still. There is a beautiful paradox here. To be alive is to be in constant motion, even in repose. Your heart, a tireless muscle, beats its rhythm hundreds of thousands of times a day, a drum calling forth life. Your diaphragm, a quiet dome of muscle, draws breath into your lungs, a gentle tide of air that sustains you. Even as your eyelids flutter or your gaze drifts across the room, it is muscle that guides these delicate adjustments. This isn't just a collection of tissues, it is a living, breathing tapestry woven from threads of incredible strength and exquisite sensitivity. To truly appreciate this, we must learn to see beyond the familiar. We must journey into the microscopic landscapes where proteins interact with the elegance of a ballet, where chemical energy transforms into mechanical work with near-perfect efficiency, and where the echoes of ancient evolutionary paths are still visible in every twitch and strain. This is a story of power and grace, of endurance and adaptation, a testament to the profound beauty of biological engineering. It's a story that rewards patient contemplation, revealing deeper layers of wonder the longer we allow our minds to drift into its intricate patterns. It is a story of you, told through the lens of the universe's most elegant machinery. Have you ever noticed the deep satisfaction that comes from a good stretch at the end of a long day? That slow, uncoiling release, a gentle invitation to your body to let go. Or perhaps the quiet strength in holding a warm cup, feeling the subtle tension in your forearm, a testament to the constant, unseen work within. These everyday sensations, so familiar they often pass unnoticed, are the surface ripples of a profound hidden ocean, the muscular system. Tonight, let us begin our quiet journey into this ocean, not with a sudden plunge, but with a gentle wade into its shallows. Let's consider the very essence of what a muscle is. It's more than just tissue that helps you move. It's a testament to life's ingenious solutions, a bridge between the unseen world of chemistry and the tangible reality of physical action. Picture, if you will, the elegant simplicity of a single purpose: to contract, to shorten, to pull. From this fundamental action springs the astonishing complexity of every movement you make, from the most powerful leap to the most delicate whisper of a smile. To truly begin, we must first understand the fundamental units, the quiet, dedicated workers. Imagine your body not as a solid block, but as a masterpiece of engineering, built from many distinct components. Among these, the skeletal muscles stand out, the ones that attach to your bones, allowing you to walk, lift, gesture, and express. Each skeletal muscle, like your biceps or your quadriceps, isn't a single, uniform mass. Instead, it's a beautifully organized bundle, a carefully woven cable of smaller and smaller units. Think of a thick rope, perhaps a mooring line on an ancient ship. That rope isn't a single strand. It's composed of many thinner ropes twisted together, and each of those thinner ropes is made of even finer fibers. Your muscle is much the same. The entire muscle is surrounded by a tough protective sheath of connective tissue, a kind of outer skin. Inside, it's divided into smaller bundles called fascicles, each wrapped in its own delicate casing. And within each fascicle we find the true marvels, the individual muscle cells or muscle fibers. These muscle fibers are not like typical cells you might imagine, small, round, and unassuming. No, muscle fibers are extraordinary. They are long, slender cylinders, sometimes stretching for the entire length of a muscle, reaching many centimeters. Imagine a single cell that is, in some cases, as long as your finger. And unlike most cells, which have a single nucleus, muscle fibers are multinucleated, containing hundreds, even thousands, of nuclei scattered along their length. This abundance of nuclei speaks to their constant activity and their need to produce vast quantities of proteins. They are like tiny biological factories humming with production. But what gives these remarkable fibers their power? If we were to peer even deeper into the heart of a single muscle fiber, we would find it packed with even tinier thread-like structures called myofibrils. These myofibrils are the true contractile elements, the actual machinery that shortens. Think of them as the individual, silken threads within our rope analogy, each one capable of pulling. They run the entire length of the muscle fiber, arranged in parallel, creating a beautiful striped pattern under a microscope, a pattern that hints at the elegant repeating units of contraction within. This intricate layering, from the whole muscle down to the myofibrils, is a testament to nature's efficiency, ensuring that every ounce of power is harnessed and directed with exquisite precision. So we've begun to unravel the macroscopic structure of a muscle, seeing it as a beautifully organized hierarchy, a nested system of bundles within bundles, culminating in the extraordinary muscle fiber. We've glimpsed the myofibrils, the fundamental threads of contraction waiting within. But how do these threads actually pull? What is the elegant dance that occurs at the most fundamental level, transforming chemical energy into the physical shortening we call movement? How do these microscopic elements, in perfect synchronicity, generate the gentle strength of a stretch or the powerful force of a leap? That question of the invisible architecture of contraction itself will be our quiet focus as we drift deeper into the heart of the muscle fiber. Welcome back, dear friends, to the tranquil halls of the Dreamy Academy. We left our previous session at the very threshold of a profound mystery. How do those incredible thread-like myofibrils within each muscle fiber actually convert chemical energy into the physical shortening we call contraction? How do they, in their silent microscopic world, orchestrate the pulling, the lifting, the very dance of life itself? Today we delve into that astonishing universe, peeling back yet another layer of this living, breathing tapestry. We venture into the heart of the contractile mechanism, where the true magic of muscular action unfolds. This will be a journey of patient contemplation, a slow and thoughtful adventure into the elegant design that underpins every gesture, every breath, every beat of our tireless hearts. Picture, if you will, the myofibril we spoke of, that long slender strand within the muscle fibre. Now, imagine that this strand is not a continuous unbroken thread, but rather a meticulously organised series of repeating units, like beads on a necklace, or perhaps more accurately, like individual self-contained engines lined up end to end. Each of these engines is a fundamental unit of contraction, a masterpiece of biological engineering known as a sarcomere. The sarcomere is the true functional core of our muscular system, the smallest contractile unit. Think of it as a meticulously designed chamber bounded at either end by dense protein-rich structures we call Z-discs. These Z-discs are like the mooring lines on an ancient ship, anchoring the entire structure, providing the framework against which the immense forces of contraction will be generated. Within the boundaries of these Z-discs lies the intricate architecture of the sarcomere, a precise arrangement of specialized proteins waiting for their cue to spring into action. Within each sarcomere we find two primary types of protein filaments, carefully interleaved and perfectly aligned: the thick filaments and the thin filaments. Their interaction is the essence of muscle contraction, a graceful and powerful dance that happens billions of times every second within our bodies. Let us first consider the thin filaments. These are primarily composed of a globular protein called actin, strung together like a double strand of pearls twisted into a helix. Imagine two delicate strings of beads spiraling round each other, forming a beautiful, intricate chain. Along this actin helix, like tiny specific docking stations, are the sites where the thick filaments will attempt to bind. But in a resting muscle, these sites are not openly available, they are guarded. Guarding these crucial binding sites are two other proteins, acting as vigilant sentinels: tropomyosin and troponin. Tropomyosin is a long, fibrous protein that wraps itself around the actin helix, effectively covering the binding sites like a protective ribbon. It's a gatekeeper, ensuring that the muscle remains relaxed until the precise moment of activation. Attached to this tropomyosin ribbon, at regular intervals, are the troponin complexes. Troponin is the key player in this regulatory dance, the molecule that will ultimately respond to the signal for contraction. Now let us turn our gaze to the thick filaments. These are made predominantly of a remarkable protein called myosin. Myosin molecules are quite distinct, often described as having a 1-1 shape. Each myosin molecule possesses a long tail and a globular head. These myosin heads are the true engines of force generation. They possess two critical capabilities. They can bind to the actin on the thin filaments, and they can also bind and hydrolyze ATP, the body's energy currency. Imagine hundreds of these myosin molecules bundled together, their long tails forming the core of the thick filament, while their globular heads protrude outwards, ready to reach for the thin filaments. These heads are like tiny biological grappling hooks, poised to engage and pull. So we have our stage set, the sarcomere, bounded by Z-discs, with interdigitating thick myosin and thin actin, tropomyosin, troponin filaments. In the resting state, the tropomyosin ribbon covers the actin binding sites, preventing the myosin heads from attaching. The muscle is relaxed, waiting. Now, how does this intricate system receive its command to contract? The signal originates from our nervous system. A nerve impulse travels down a motor neuron and reaches the muscle fiber. This electrical signal triggers the release of a crucial chemical messenger, which in turn causes a sudden surge of calcium ions, Ca2+, into the sarcoplasm, the cytoplasm of the muscle cell. These calcium ions are stored within a specialized network of membranes surrounding the myofibrils, known as the sarcoplasmic reticulum, a kind of internal reservoir ready to release its precious cargo upon command. And here, dear friends, is where the true wonder of the sliding filament theory unfolds. This theory, a cornerstone of our understanding of muscle physiology, explains how these filaments interact to generate force. When those calcium ions flood into the sarcoplasm, they don't simply float aimlessly. They are drawn with exquisite specificity to the troponin molecules on the thin filaments. Remember troponin, the sentinel attached to tropomyosin. When calcium binds to troponin, it causes a subtle but profound change in troponin's shape. This conformational shift is the first domino to fall. Because troponin is intimately linked to tropomyosin, its change in shape tugs on the tropomyosin ribbon. This tugging action effectively pulls the tropomyosin away from the actin binding sites, exposing them. It's like drawing back a curtain, revealing the stage where the myosin heads are waiting. With the binding sites on actin now exposed, the myosin heads, which have been cocked or energized by the hydrolysis of ATP, though still holding on to ADP and an inorganic phosphate group PI, are free to attach. This attachment forms what we call a cross bridge, a physical link between the thick and thin filaments. Once the cross bridge is formed, a remarkable event occurs, the power stroke. The myosin head, having released its ADP and PI, pivots and bends, pulling the thin filament along with it towards the center of the sarcomere. Imagine that golf club head swinging, not to hit a ball, but to tug on the actin filament. This is the very essence of muscle contraction. The myosin heads are not shortening themselves, but rather pulling the actin filaments past them. The filaments are sliding, not shortening. After the power stroke, the myosin head remains attached to actin, but now in a low-energy state. To detach and prepare for another cycle, a fresh molecule of ATP must bind the myosin head. The binding of ATP causes the myosin head to release from the actin. This is a crucial step. Without ATP, the myosin heads cannot detach, leading to the sustained contraction we observe in rigor mortis. Once detached, the ATP molecule is immediately hydrolyzed by the myosin head into ADP and pi. This hydrolysis re-energizes the myosin head, causing it to recock or return to its high-energy position, ready to bind to another exposed site on the actin filament further down the line. This entire cycle, calcium binding, tropomyosin shift, myosin binding, power stroke, ATP binding, detachment and recocking, repeats itself as long as calcium ions are present and ATP is available. Each power stroke pulls the thin filaments a tiny increment closer to the center of the sarcomere. As countless myosin heads perform this synchronized pulling action, across all the sarcomeres within a myofibril, and across all the myofibrils within a muscle fiber, the entire muscle fiber shortens. Consider the sheer elegance of this mechanism. It's not a sudden, violent yank, but a series of rapid, coordinated, tiny tugs. It's like a team of rowers, each pulling their oar in sequence, propelling the boat forward. The cumulative effect of billions of these microscopic power strokes is the macroscopic movement we observe in our bodies, from the subtle flick of an eyelid to the powerful leap of an athlete. What happens when the signal to contract stops? When the nerve impulse ceases, the calcium ions are rapidly pumped back into their storage sites within the sarcoplasmic reticulum. This active pumping requires ATP, highlighting the constant energy demands of muscle function, even in relaxation. With the calcium ions removed from the sarcoplasm, they detach from troponin. Troponin then reverts to its original shape, allowing the tropomyosin ribbon to slide back into place, once again covering the actin-binding sites. The myosin heads can no longer form cross bridges, and the muscle relaxes, returning to its resting length. This intricate dance of proteins, the precise choreography of calcium and ATP, is nothing short of miraculous. It is a testament to the astonishing efficiency and complexity of biological systems. Every time you lift a cup of tea, every time you take a step, every time your heart beats, this microscopic ballet is unfolding within you, a silent symphony of molecular machines. We began this journey by marveling at the macroscopic architecture of muscle, its bundles within bundles. Today we have zoomed into the very heart of its function, to the sarcomere, and uncovered the ingenious mechanism by which it transforms an electrical signal into mechanical force. It is a system of breathtaking precision, capable of both immense power and exquisite delicacy. As we conclude our session today, take a moment to truly appreciate the incredible, invisible work happening within your own body right now. Feel the subtle tension in your posture, the gentle rise and fall of your chest with each breath. All of this, and so much more, is powered by the relentless, elegant sliding of filaments within billions of tiny sarcomeres. It is a quiet marvel, a profound testament to the living, breathing wonder that is the human form. Next time we will explore the energy systems that fuel this incredible machinery, delving into how our cells generate the vast quantities of ATP required for continuous muscular activity. Until then, may your contemplations be peaceful, and your appreciation for the silent architects of your motion grow ever deeper. Thank you for joining us in the Dreamy Academy. The intricate dance of actin and myosin, the precise choreography of cross-bridge formation and power stroke, the elegant disengagement facilitated by a fresh ATP molecule. These are the heartbeats of muscle contraction. We've journeyed deep into the sarcomere, witnessing the sliding filaments in their silent, tireless work. But this work, this perpetual motion, demands an incessant supply of energy. Just as a meticulously crafted machine requires fuel to perform its function, so too do our muscles depend on a constant, readily available source of ATP. It is ATP, after all, that is the direct currency of life, the immediate fuel for every cellular endeavour. Without it, the graceful ballet of muscle contraction grinds to a halt. Today, we turn our patient contemplation to the astonishing universe of our body's energy systems, the ingenious mechanisms that tirelessly regenerate ATP, ensuring that our muscles, from the subtlest twitch to the most monumental lift, are always powered. It's a story of remarkable efficiency, of chemical transformations that allow us to run, to breathe, to simply hold a pen. Picture, if you will, a vast complex city. Just as a city requires various power plants, some for immediate high-demand needs, others for steady long-term supply, our muscles possess a sophisticated network of energy factories, each specialized for different demands. Our journey begins with the sprint, the sudden burst of power, the immediate explosive demand for energy. Have you ever noticed how you can exert maximum effort for only a very short duration, a few seconds perhaps, before your muscles begin to falter? This immediate potent energy comes from a system that is both remarkably fast and incredibly limited. The phosphocreatine system. Imagine your muscle cells as having a small, highly efficient emergency battery, always charged and ready for instant deployment. This battery is phosphocreatine, also known as creatine phosphate. It's a high-energy phosphate compound stored right within the muscle fibers, a chemical cousin to ATP itself. When the muscle first contracts, when that initial surge of ATP is rapidly consumed by the myosin heads, the cell doesn't wait for complex metabolic pathways to kick in. Instead, it turns to this immediate reserve. The enzyme creatine kinase acts as the swift conductor of this emergency transfer. In a fraction of a second, it snatches a phosphate group from phosphocreatine and rapidly attaches it to a molecule of ADP, adenosine diphosphate, instantaneously converting it back into ATP. It's a chemical sleight of hand, a direct and rapid rephosphorylation that bypasses the need for oxygen or the breakdown of glucose. Consider for a moment the sheer elegance of this system. When you need to escape danger, lift a heavy object unexpectedly, or launch into a maximal jump, your body doesn't pause to prepare. The phosphocreatine system provides that initial explosive burst of power, fueling the first 6 to 10 seconds of intense activity. It's the ultimate short-term solution, a crucial survival mechanism that allows for immediate high-power output. But, like any emergency battery, its charge is quickly depleted. Once the phosphocreatine stores are exhausted, the muscle must turn to other, more substantial energy sources. As the phosphocreatine system wanes, and if the demand for ATP remains high, our muscles transition to the next power plant in our cellular city, glycolysis or anaerobic respiration. This system steps in when the immediate reserves are gone, but oxygen supply isn't sufficient to meet the rapid ATP demand. Glycolysis is a beautiful ancient metabolic pathway, a testament to life's earliest forms. It's the breakdown of glucose, our body's preferred sugar, into a simpler compound called just pyruvic acid. This process occurs in the cytoplasm of the muscle cell, outside the mitochondria, and crucially, it does not require oxygen. This is why we call it anaerobic. Picture a small, bustling factory operating at high speed. Glucose molecules derived either from the blood or from the muscle's own stored glycogen, a complex carbohydrate chain, like a linked string of glucose beads, enter this factory. Through a series of 10 enzymatic reactions, each a precise step, the glucose molecule is broken down. The net result of this frantic activity is the production of a modest amount of ATP, just 2 molecules for every molecule of glucose, and 2 molecules of pyruvic acid. While 2 ATPs might seem like a small yield compared to what we'll discuss next, the speed of glycolysis is its superpower. It can generate ATP much faster than oxygen-dependent pathways, making it ideal for activities that last from roughly 10 seconds up to about 2 minutes. Think of a 400-meter sprint or a few intense sets of weightlifting. Here, glycolysis is the dominant provider, churning out ATP to sustain the effort. However, this rapid production comes with a trade-off. In the absence of sufficient oxygen, which is often the case during intense sustained activity, the pyruvic acid produced by glycolysis cannot enter the mitochondria for further, more efficient breakdown. Instead, it gets converted into lactic acid. This lactic acid then rapidly dissociates into lactate and hydrogen ions. It is the accumulation of these hydrogen ions that leads to a decrease in pH within the muscle cells, interfering with enzymatic activity and muscle contraction. This is what we often perceive as the burning sensation and fatigue during strenuous exercise. It's the body's warning system, signaling that the anaerobic factory is working overtime and producing a metabolic byproduct that needs to be cleared. So while glycolysis offers a faster, more substantial ATP supply than the phosphocreatine system, it's not sustainable indefinitely due to its limited ATP yield per glucose molecule and the accumulation of metabolic byproducts. It's a bridge, a vital link between the immediate burst and the long-haul fuel. And for that long haul, for sustained activity, for the quiet endurance that defines so much of our daily lives, we turn to the most powerful and efficient of our energy systems, oxidative phosphorylation, or aerobic respiration. This is the grand symphony of energy production, requiring the presence of oxygen, and it takes place within the cellular powerhouses we know as mitochondria. Imagine these mitochondria as miniature, highly sophisticated power stations, equipped to process a variety of fuels with incredible efficiency. Unlike glycolysis, which only uses glucose, aerobic respiration can break down not just glucose, but also fats, fatty acids, and, in extreme cases, even proteins, amino acids, to generate ATP. This adaptability makes it the primary energy system for activities of moderate to low intensity that last for more than a few minutes, from walking and jogging to reading a book, or simply maintaining your posture. The pyruvic acid generated from glycolysis, when oxygen is abundant, enters the mitochondria. Here, it is further broken down in a cyclical series of reactions known as the Krebs cycle, or citric acid cycle. This cycle doesn't directly produce a huge amount of ATP, but it generates electron carriers, NADH and FADH2, which are crucial for the final, most productive stage. These electron carriers then feed into the electron transport chain, a series of protein complexes embedded in the inner mitochondrial membrane. This is where the magic truly happens. As electrons are passed down this chain, energy is released, which is used to pump hydrogen ions across the membrane, creating a gradient. This gradient then drives the synthesis of a massive amount of ATP, up to 30 to 32 molecules of ATP per molecule of glucose, a stark contrast to the mere 2 from glycolysis. This process also utilizes oxygen as the final electron acceptor, producing water as a byproduct, and carbon dioxide is released during the Krebs cycle. The sheer efficiency of aerobic respiration is breathtaking. It provides a steady, copious supply of ATP, allowing for prolonged activity without the rapid onset of fatigue caused by lactic acid accumulation. It's the system that allows us to run marathons, to sustain complex thoughts, to live our lives. It's slower to kick in compared to the other systems, requiring a few minutes to ramp up to full capacity as oxygen delivery and mitochondrial activity increase. But once engaged, it is a tireless, clean burning engine, These three systems, the rapid but limited phosphocreatine system, the faster but less efficient anaerobic glycolysis, and the slower but immensely powerful aerobic respiration, do not operate in isolation. They are beautifully integrated, constantly adjusting their contributions based on the intensity and duration of the muscular effort. When you begin to exercise, the phosphocreatine system provides the initial burst. As this depletes, glycolysis quickly takes over, providing energy for the next minute or two. Simultaneously, the aerobic system is slowly mobilizing, increasing oxygen uptake and mitochondrial activity. If the activity is low to moderate intensity, the aerobic system will eventually become the dominant ATP provider, capable of sustaining the effort for hours. If the intensity remains high, glycolysis continues to dominate, leading to lactate accumulation and eventual fatigue. Consider the incredible adaptability of this biological engineering. A sprinter relies heavily on the phosphocreatine system and anaerobic glycolysis for their explosive short-duration effort. A marathon runner, on the other hand, depends almost entirely on aerobic respiration, efficiently burning fats and carbohydrates over extended periods. Even a simple gesture, like lifting your hand to wave, involves a rapid, seamless interplay of these systems, orchestrated with exquisite precision. This intricate interplay is also reflected in the very design of our muscle fibers. Some fibers are specialized for rapid, powerful, short-duration contractions, rich in phosphocreatine and glycolytic enzymes, fast-twitch fibers. Others are built for endurance, packed with mitochondria and myoglobin, an oxygen-binding protein giving them a reddish hue, optimized for aerobic respiration, slow-twitch fibers. We'll delve deeper into these fascinating cellular specializations in our next session. For now, let us simply marvel at the quiet genius within. The constant demand for ATP, met by a sophisticated layered energy supply system, ensures that our muscles are always ready for action. From the cellular machinery that pulls thin filaments across thick, to the metabolic pathways that churn out the energy currency, our bodies are a living, breathing tapestry of interconnected wonders. Each breath we take, each beat of our heart, each thought we ponder, is a testament to the tireless work of these tiny biological factories silently fueling the astonishing universe within. It is truly a masterpiece of engineering, a testament to the patient contemplation of life itself. We've journeyed through the intricate dance of contraction and explored the energetic fuel stations that power our every move. We've seen how ATP, that tiny currency of life, is meticulously generated and spent, adapting to the demands placed upon our living, breathing tissues. But as we consider the vast spectrum of human activity, from the gentle, sustained sway of a dancer to the explosive leap of a high jumper, a question naturally arises: how can the same muscle tissue perform such wildly different feats with such remarkable precision? The answer, dear friends, lies in a profound and elegant specialization, a testament to the body's astonishing efficiency, the existence of distinct muscle fiber types. Imagine, if you will, that our muscles are not a monolithic entity, but rather a diverse team, each member uniquely suited to a particular task. This is where our quiet exploration takes us today, into the nuanced world of muscle fiber differentiation, where we uncover the specific characteristics that allow our muscles to be both marathon runners and sprinters simultaneously. As we delve into this, I invite you to picture the fibers themselves, not as abstract concepts, but as living threads, each with its own story, its own particular way of contributing to the grand symphony of motion. We primarily categorize these fibers into two broad families, each with its own unique physiological signature. Slow-twitch fibers, often referred to as type 1, and fast-twitch fibers, which encompass type 2A and type 2X. Let us begin with the slow-twitch fibers, the patient, unwavering flames within our musculature. These are the quiet workhorses, the endurance champions, designed for sustained, low-intensity efforts. If you've ever held a posture for an extended period, or walked for miles without conscious thought, you've been relying heavily on these remarkable fibers. Visually, if we were to peer inside, type 1 fibers appear reddish. This isn't merely an aesthetic detail, it's a profound clue to their function. The rich, ruby hue comes from a high concentration of myoglobin, a protein akin to hemoglobin that stores oxygen within the muscle cell itself. Think of myoglobin as a personal oxygen reservoir, ready to release its precious cargo to the mitochondria when needed. And speaking of mitochondria, these fibers are absolutely brimming with them. Tiny biological power plants specialized in aerobic respiration. They are also lavishly supplied with capillaries, ensuring a constant, fresh delivery of oxygen and nutrients, and efficient removal of waste. Physiologically, these characteristics translate into a slow contraction speed. While they don't generate immense power in a single burst, their true strength lies in their unparalleled resistance to fatigue. They are the epitome of efficiency, utilizing oxidative phosphorylation to continuously churn out ATP, making them ideal for activities that require stamina rather than explosive force. Consider the muscles responsible for maintaining your posture throughout the day, the quiet, constant adjustments that keep you upright. These are predominantly slow-twitch fibers, performing their tireless duty hour after hour without complaint. They are the steady beat of the drum, the deep and resonant hum that underlies all our movements. Now, let us turn our attention to the more dynamic, more explosive members of our muscular team, the fast-twitch fibers. As their name suggests, these fibers are built for speed and power, for those moments when a sudden, forceful effort is required. Unlike their slow-twitch counterparts, fast-twitch fibers are designed for rapid, powerful contractions, though often at the cost of endurance. Within the fast-twitch family, we further distinguish between two primary subtypes, each representing a slightly different specialization. Type A, fast oxidative glycolytic, and type 2, fast glycolytic. The type 2 fibers are in many ways a fascinating hybrid, a bridge between the endurance of slow twitch and the power of fast twitch. They possess a remarkable versatility, capable of both aerobic and anaerobic metabolism. If you were to observe them, they would appear pinkish or reddish, reflecting a respectable amount of myoglobin and mitochondria, though less than type 1, and a decent capillary network. They contract faster and generate more force than type 1 fibers, and while they are more fatigable than slow twitch, they possess a greater resistance to fatigue than their purely glycolytic fast twitch cousins. Think of a middle-distance runner who needs both speed and the ability to sustain that speed for a significant duration. Type 2 fibers are their secret weapon, allowing for powerful bursts that can also be maintained for a moderate length of time. They are the versatile athletes, capable of adapting to many different challenges. Then we arrive at the type 2X fibers, the true powerhouses, the sprinters and weightlifters of our muscular landscape. These are the fibers built for sheer, unadulterated explosive force. Visually, they are the palest of the bunch, appearing whitish. This lack of colour is again a key indicator of their physiology. They have very low concentrations of myoglobin, fewer mitochondria, and a sparser capillary supply. Their primary mode of ATP production is anaerobic glycolysis, which, as we discussed, is incredibly fast but also leads to rapid fatigue and the accumulation of lactic acid. Type 2X fibers contract with immense speed and generate the greatest amount of force. However, they are also the most easily fatigable. They are designed for these fleeting moments of maximum effort, the single powerful jump, the sudden sprint, the heavy lift. They are the lightning strike, brilliant and potent, but brief. Imagine a short-distance sprinter exploding from the blocks, or a powerlifter executing a maximum lift. These actions are powered predominantly by the rapid, forceful contractions of type 2X fibers, which ignite with incredible intensity, but quickly exhaust their limited anaerobic fuel reserves. It's important to understand that no muscle is composed of only one type of fiber. Rather, most muscles contain a mixture of all three types, though the precise ratio will vary depending on the muscle's primary function. For instance, postural muscles in the back tend to have a higher proportion of slow-twitch fibers, while muscles involved in powerful, quick movements, like those in the arms and legs, will have a greater percentage of fast-twitch fibers. This brings us to another fascinating aspect of muscular specialization: motor unit recruitment, often described by Henneman's size principle. When our nervous system decides to initiate a movement, it doesn't just activate all muscle fibers at once. Instead, it follows a precise economical order. For low-force activities, like simply lifting a pen, the brain first recruits the smallest motor units, which are typically composed of slow-twitch type 1 fibers. These are the most energy-efficient and fatigue-resistant. As the demand for force increases, say you pick up a heavier book, the nervous system progressively recruits larger motor units, which include the type 2A fibers. And for those moments of maximum effort, when you need to lift something truly heavy or sprint at full speed, the largest motor units, comprising the powerful type 1 IX fibers, are finally called into action. Picture it like turning up the volume on a stereo. You start with a gentle hum, then gradually increase the sound as needed, rather than blasting it at full volume from the start. This orderly recruitment allows for smooth, graded control of muscle force, conserving energy when only a small effort is required, and unleashing full power only when absolutely necessary. It's a symphony of finely tuned control, orchestrated by the nervous system. Now, you might be wondering about the role of genetics versus training in determining our fiber type composition. The general consensus is that our proportion of slow twitch versus fast twitch fibers is largely genetically predetermined. Some individuals are naturally predisposed to be endurance athletes due to a higher percentage of slow twitch fibers, while others are born with a greater proportion of fast twitch fibers, making them naturally inclined towards power and speed sports. However, this doesn't mean we are entirely bound by our genetic blueprint. The body, in its infinite wisdom, possesses a remarkable capacity for adaptation. while you can't fundamentally change a type 1 fiber into a type 2X, training can induce significant changes within fiber types. For example, consistent endurance training can enhance the oxidative capacity of all fiber types, making type 2A fibers behave more like type 1 in terms of fatigue resistance. Conversely, resistance training can increase the size and strength of fast-twitch fibers, making them even more powerful. It's a beautiful dance between nature and nurture, where our inherent potential is shaped and refined by the challenges we present to our bodies. So, as we bring this exploration of muscle fiber types to a close, let's take a moment to appreciate the sheer ingenuity of this biological design. Our muscles are not just bundles of tissue, they are sophisticated, adaptable engines, each fiber type a specialized component, contributing to the incredible range of human movement. From the subtle, unconscious adjustments that maintain our posture, to the explosive, gravity-defying feats of an athlete, it is this precise specialization that allows our bodies to navigate the world with such grace, power, and enduring resilience. In our next session, we will shift our focus from the individual fiber to the larger systems that control them, delving into the fascinating world of motor units and how the nervous system orchestrates these diverse teams of fibers to create coordinated, purposeful movement. Until then, I invite you to contemplate the silent, tireless work of your own muscle fibers, marveling at the incredible engineering that allows you to simply be. And so, we arrive at a pivotal juncture in our peaceful exploration, a moment where the silent architects of motion meet their brilliant conductor. We've journeyed deep into the muscle fiber, witnessed the intricate dance of the sarcomere, understood the tireless energy systems that fuel its every beat, and marveled at the specialized roles of slow and fast-twitch teams. Yet, all this incredible machinery, this living, breathing tapestry of power and grace, would lie dormant, an inert masterpiece, were it not for the exquisite whispers and precise commands of the nervous system. Today, we turn our gaze to this profound partnership, this ceaseless conversation between nerve and muscle, which orchestrates every conscious intention and every unconscious reflex that defines our movement. We will unravel the delicate threads that connect thought to action, exploring how the brain, with breathtaking precision, recruits, energizes, and fine-tunes the vast army of muscle fibers to achieve a symphony of motion. Imagine your body not just as a collection of parts, but as a grand orchestra. The muscles are the diverse sections, the powerful brass, the nimble strings, the rhythmic percussion. But what good is an orchestra without a conductor? The nervous system is that maestro standing at the podium, interpreting the score of our intentions and bringing each section, each individual player, into perfect harmony. At the heart of this orchestration lies a fundamental unit, one we touched upon briefly in our discussion of fiber types, but which now demands our full, contemplative attention: the motor unit. Picture it as the smallest, most indivisible command structure in our muscular army. A single motor neuron, a specialized nerve cell extending from the spinal cord, reaches out and branches, its tendrils innervating a specific group of muscle fibers. These fibers, all of the same type, be it slow twitch, type 2A, or type 2X, respond as one. When that motor neuron fires, all the muscle fibers it commands contract simultaneously. It's a beautifully simple, yet profoundly effective principle of synchronized action. Consider for a moment the implications. A single motor neuron might innervate just a handful of muscle fibers in a muscle, requiring fine, delicate control, like those moving your eyeballs or fingers. This grants us exquisite precision. Conversely, a motor neuron might command hundreds, even thousands, of fibers in large, powerful muscles like those in your quadriceps, where brute force is often paramount. This varying ratio of nerve to muscle fiber is a testament to the body's astonishing efficiency and adaptability, tailored perfectly to the specific function of each muscle. Now, let us zoom in even further to the very point of contact, the sacred space where the nervous system's electrical language is translated into the muscle's mechanical action. This is the neuromuscular junction, NMJ, a synaptic whisper, a molecular handshake of profound significance. Imagine the terminal end of our motor neuron, swollen and rich with tiny vesicles, like miniature balloons filled with a precious chemical messenger. This is the presynaptic terminal. It hovers over a specialized region of the muscle fiber membrane, known as the motor end plate, which is intricately folded, increasing its surface area like a delicate, convoluted landscape. Between these two, a microscopic chasm, the synaptic cleft, acts as a silent, expectant void. When an electrical signal, an action potential, races down the motor neuron, it arrives at the presynaptic terminal. This arrival triggers a cascade of events. Voltage-gated calcium channels in the nerve terminal open, allowing a rush of calcium ions inward. This influx is the signal for those tiny vesicles to fuse with the presynaptic membrane, spilling their contents into the synaptic cleft. The precious cargo they release is our key neurotransmitter for muscle contraction, acetylcholine, ACHS, liberated into the cleft, diffuses swiftly across this tiny gap, drawn to specialized receptors embedded within the folds of the motor end plate on the muscle fiber. When ACH binds to these receptors, it's like a key turning a lock, causing ion channels to open. Sodium ions, eager to enter, rush into the muscle fiber, while potassium ions slowly exit. This rapid influx of positive charge causes a localized depolarization of the muscle membrane, known as an end plate potential. If this potential reaches a critical threshold, it triggers a full-blown action potential that sweeps across the entire surface of the muscle fiber, much like a ripple expanding across a pond. This muscle action potential is the crucial link, the baton pass from the electrical language of the nerve to the contractile machinery of the muscle. We recall from Chapter 2 how this electrical signal then dives deep into the muscle fiber via the T-tubules, signaling the sarcoplasmic reticulum to release its stored calcium ions. It is these calcium ions binding to troponin that shift tropomyosin, exposing actin's binding sites, allowing myosin heads to form cross bridges and initiate the power stroke. Thus, the nerve's command, born from a thought or a reflex, is meticulously translated into the physical shortening of the sarcomere, the very essence of muscle contraction. But how does the nervous system, our masterful conductor, fine-tune the strength of a muscle contraction? How do we lift a feather with delicate grace, yet summon immense power to lift a heavy weight? The answer lies in two elegant mechanisms: motor unit recruitment and the frequency of stimulation. We've already introduced motor unit recruitment in Chapter 4, but let us now appreciate it from the conductor's perspective. The brain doesn't simply command, quote, 5 and expect all muscle fibers to fire at once. Instead, it employs a sophisticated, graded approach, often referred to as Henneman's size principle. For weak, precise movements, the brain first activates the smallest motor units, those innervating only a few muscle fibers, typically slow twitch, type Y fibers. These are the most easily excitable neurons, requiring the least amount of stimulus to fire. They provide a low, sustained level of force, ideal for maintaining posture or performing delicate tasks. As the demand for force increases, the brain progressively recruits larger motor units. Next come the motor units controlling type 2A, fast oxidative glycolytic fibers, offering more power and speed. Finally, for maximum effort, for those explosive bursts of strength, the largest motor units, those commanding the powerful type 2X, fast glycolytic fibers, are brought into play. This systematic, orderly recruitment allows for a smooth, finely graded increase in muscle tension, preventing jerky, all-or-nothing movements. It's like a dimmer switch, gradually increasing the light rather than a simple on-off switch. Our conductor brings in the quiet violins first, then adds the cellos, and only when a crescendo is needed the full brass section. The second mechanism, equally vital, is the frequency of stimulation. Imagine sending a single, isolated electrical pulse down a motor neuron. This results in a brief, weak contraction called a first muscle twitch. The muscle contracts, then fully relaxes, but what if the nerve sends a second stimulus before the muscle has completely relaxed from the first? The second contraction will be stronger than the first, adding its force to the residual tension of the previous twitch. This phenomenon is known as wave summation. The muscle doesn't have time to fully return to its resting state, so the subsequent stimuli build upon the previous ones, increasing the overall force. As the frequency of stimulation increases further, the individual twitches begin to fuse, creating a sustained but still slightly wavering contraction, known as unfused or incomplete tetanus. The muscle is contracting continuously, but you can still detect the slight relaxation periods between stimuli. Push the frequency even higher, and the twitches completely fuse into a smooth, sustained, maximal contraction, called fused or complete tetanus. This is the state where the muscle is producing its greatest possible force, and it is this smooth, sustained contraction that allows for the fluid, coordinated movements we perform every day, from walking to writing to playing a musical instrument. Without this precise control over stimulation frequency, our movements would be a series of jerky, isolated twitches, utterly lacking in grace and effectiveness. Even when we are at rest, our muscles are never entirely silent. A subtle, continuous state of partial contraction, maintained by the asynchronous firing of a small number of motor units, pervades our musculature. This is muscle tone, a quiet vigilance, a constant readiness. It keeps our muscles slightly firm, prepared for action, and plays a crucial role in maintaining posture, stabilizing joints, and generating the heat that helps regulate our body temperature. It's the hum of an engine at idle, ready to accelerate at a moment's notice. But the nervous system's role isn't merely about sending commands, it's also about listening. To effectively coordinate movement, the brain needs constant feedback about the state of our muscles, their length, their tension, their position in space. This vital sensory information is provided by specialized receptors within the muscles and tendons, collectively known as proprioceptors. They are our body's internal GPS, constantly updating the brain on our physical status. Two types of proprioceptors are particularly important. The first are muscle spindles. Tiny, intricate structures embedded within the belly of the muscle itself, running parallel to the main muscle fibers. These spindles are exquisitely sensitive to changes in muscle length and the rate of change of that length. When a muscle is stretched, the muscle spindles are also stretched, sending a barrage of signals to the spinal cord and brain. This information is critical for maintaining posture and for the air stretch reflex, a protective mechanism that causes a stretched muscle to contract, preventing overstretching and potential injury. Think of the patellar reflex, where a tap below the kneecap stretches the quadriceps, causing it to contract and kick the leg forward. The second type of proprioceptor we consider today are the Golgi tendon organs, GTOS, located within the tendons. Where muscle transitions into bone, these receptors are sensitive to changes in muscle tension. When a muscle contracts forcefully, or when tension is applied to the tendon, the GTOs are activated. They send inhibitory signals to the motor neuron supplying that muscle, causing it to relax. This tendon reflex acts as another protective mechanism, preventing muscles from generating excessive force that could damage the muscle or tear the tendon. It's a built-in safety switch, ensuring that our pursuit of strength doesn't lead to self-harm. Thus, the nervous system and the muscular system engage in a constant, intricate dialogue. The brain sends commands down the motor neurons, initiating contraction. Simultaneously, the proprioceptors send a rich stream of sensory information back to the brain, detailing the muscle's response. This continuous feedback loop allows the nervous system to make real-time adjustments, ensuring that our movements are not only initiated but also smoothly executed, finely controlled, and safely performed. We have traversed from the grand vision of muscle fibers to the microscopic dance of neurotransmitters, from the subtle art of recruitment to the protective wisdom of reflexes. We have seen how the nervous system, with its astonishing complexity and precision, acts as the ultimate orchestrator, transforming our intentions into the beautiful, dynamic reality of movement. The human body, in its ability to coordinate such diverse and specialized components into a seamless whole, stands as an enduring testament to the marvel of biological engineering. And as we conclude this chapter, I invite you to carry this sense of wonder into your everyday life, observing the silent, intricate ballet unfolding within you with every breath, every gesture, every step. It is a profound and ongoing revelation. We've journeyed deep into the microscopic world, unraveling the intricate dance of filaments and the tireless work of ATP. We've marveled at the nervous system, the maestro conducting this internal symphony, receiving constant feedback from our proprioceptors, those quiet sentinels of position and tension. Now, let us step back and consider the macroscopic marvel that emerges from this exquisite detail, movement itself. How do these meticulously orchestrated contractions translate into the graceful arc of an arm, the powerful thrust of a leg, or the subtle stability that keeps us upright against the ceaseless pull of gravity? Today, we turn our gentle gaze to the different ways muscles contract and how they collaborate in a silent, continuous ballet to sculpt our every action. Imagine your body, not as a collection of isolated parts, but as a living, breathing sculpture in constant flux, every posture held, every object lifted, every step taken, is a testament to the nuanced language of muscle contraction. And within this language, there are two primary dialects we must understand: isometric and isotonic contractions. Consider for a moment the simple act of holding a heavy book. Your arm is outstretched, unmoving. The book remains suspended. Is your biceps muscle working? Absolutely. It is generating force, a significant amount to counteract the downward pull of gravity on the book. Yet, the length of your biceps muscle is not visibly changing. This, my friends, is the essence of an symmetric contraction. Iso meaning same and metric referring to length. In an isometric contraction, the muscle generates tension. It tries to shorten, but the external resistance is equal to or greater than the force it can produce, preventing any change in its overall length. Think of it as a silent, unwavering standoff. Your muscles are like the mooring lines on an ancient ship, taut and strong, holding it against the tide even as the ship itself remains stationary. This type of contraction is fundamental to our stability. It's how we maintain posture, how we brace ourselves, how we keep our head upright as we listen. It's the quiet, often unseen work that provides the foundation for all dynamic movement. Without isometric contractions, we would be like a marionette with slack strings, collapsing into ourselves. It is the art of holding, of resisting, of maintaining a silent, powerful equilibrium. Have you ever noticed the subtle trembling in your muscles when you hold a difficult yoga pose or carry a heavy grocery bag for too long? That trembling is the visible manifestation of countless motor units engaging and disengaging in a tireless isometric effort, each attempting to maintain that same length against an insistent load. It's a profound testament to the body's unwavering commitment to stability. Now, let us introduce the other primary dialect, isotonic contractions. Here, iso still means same, but tonic refers to tension. While it's a bit of a misnomer, tension does fluctuate throughout an isotonic movement. The defining characteristic is that the muscle changes length while generating force, overcoming the external resistance. This is where the magic of visible movement truly unfolds. Within isotonic contractions, we discern two beautiful complementary phases: concentric and eccentric. Picture, if you will, the act of lifting that same heavy book from a table to your chest. As your biceps muscle shortens, pulling your forearm upwards, it is performing a concentric contraction. The muscle's force generation exceeds the resistance, causing it to visibly shorten and the joint angle to decrease. This is what most of us instinctively think of when we imagine a muscle working, the powerful upward surge, the overcoming of gravity. It is the active, triumphant phase where the muscle is quite literally pulling the world closer or pushing it away. Think of a dancer leaping, a climber pulling themselves up a rock face, or a child reaching for a toy. These are moments of concentric power, where the muscle, through its shortening, dictates the direction and velocity of movement. But what happens when you lower that book back to the table? Do you just let it drop? Of course not. You control its descent, a slow, deliberate return. As your biceps muscle lengthens, resisting the pull of gravity, it is performing an eccentric contraction. Here, the external resistance exceeds the force the muscle is generating, causing the muscle to lengthen under tension. This is a truly remarkable feat of biological engineering, Often overlooked yet profoundly important, eccentric contractions are like a controlled surrender, a graceful deceleration. They are the brakes of our muscular system, allowing us to absorb impact, to lower objects gently, to control the descent during a squat, or to land softly after a jump. Paradoxically, eccentric contractions can generate even greater force than concentric ones, and they are often responsible for much of the muscle soreness we feel after a new or intense workout, as they place unique demands on the muscle fibers, causing microtrauma that ultimately leads to adaptation and strength gains. It is the art of resisting, of yielding with power, of gracefully managing the forces that constantly act upon our bodies. So, in every single movement, from the simplest gesture to the most complex athletic feat, we see this intricate interplay. Isometric contractions providing stability, concentric contractions generating power and movement, and eccentric contractions controlling and decelerating. They are not isolated events, but rather a continuous flowing conversation between our muscles and the world around us. Now let's expand our perspective even further. No muscle acts in isolation. Our bodies are not a collection of independent levers, but a finely tuned orchestra, where each muscle plays its specific role to create a harmonious symphony of movement. To truly appreciate this, we must understand the concept of muscle interactions. When we decide to perform a specific action, say bending our elbow, one muscle takes the lead. This is the agonistine or the stem, prime mover. For our elbow bend, the biceps brachii is a classic example of an agonist. It is the primary muscle responsible for initiating and executing that specific movement. But for the biceps to effectively shorten and pull, its opposing muscle, the antagonist, must either relax or lengthen in a controlled manner. In the case of elbow flexion, the triceps brachii, located on the back of the arm, is the antagonist. If both the biceps and triceps contracted forcefully at the same time, the arm would lock up, unable to move. This coordinated relaxation or controlled lengthening of the antagonist is just as crucial as the agonist's contraction. It's like a dancer extending their leg. The muscles on the front of the thigh contract, while those on the back must release their tension to allow the full extension. The antagonist, therefore, is not an enemy, but a crucial partner, providing balance and control. Beyond the agonist and antagonist, we have other vital players, the synergists. These muscles assist the agonist, either by adding extra force to the movement or, more commonly, by preventing unwanted movements. Imagine trying to make a tight fist. While the muscles in your forearm are the prime movers for finger flexion, the synergists in your wrist prevent your wrist from flopping back, ensuring that the force is directed efficiently to your fingers. They are the subtle helpers, the fine tuners, ensuring precision and efficacy in our movements. And then we have the calf fixators. These muscles specialize in stabilizing the origin of the prime mover, or stabilizing a joint that is not directly involved in the movement, but whose stability is essential for the prime mover to act effectively. Think of your shoulder muscles, like the rotator cuff, when you lift something heavy with your arm. While your biceps might be the agonist for lifting, your shoulder muscles act as fixators, anchoring your arm to your torso, providing a stable base from which your biceps can pull. Without these fixators, the force generated by the agonist would be dissipated, and the movement would be weak or uncontrolled. They are the silent anchors, the unmoving foundations that allow the dynamic action to flourish. So, every time you reach for a cup, kick a ball, or even just shift your weight, a complex orchestral performance is underway. Agonists lead, antagonists yield, synergists assist, and fixators stabilize, all in a seamless, subconscious coordination that is nothing short of miraculous. This intricate collaboration is a testament to the body's holistic design, where every part contributes to the greater whole. This brings us naturally to the fascinating interaction of muscles with our skeletal framework. Our bones, far from being inert structures, act as a system of levers, allowing muscles to generate movement, amplify force, or increase speed. A lever, in its simplest form, consists of a rigid bar, our bone, a fulcrum, our joint, a force applied by our muscle, and a resistance to be overcome, the weight of our limb or an object we are moving. The human body employs 3 classes of levers, each designed for specific mechanical advantages. First-class levers are like a seesaw, with the fulcrum positioned between the effort and the resistance. Think of lifting your head off your chest. Your neck muscles provide the effort, your atlanto-occipital joint is the fulcrum, and the weight of your head is the resistance. They can be designed for balance, speed, or force, depending on the relative positions. Second-class levers place the resistance between the fulcrum and the effort. Imagine standing on your tiptoes. The ball of your foot is the fulcrum, your body weight is the resistance, and your calf muscles provide the effort. These levers are built for power, allowing a smaller effort to move a larger resistance, though often at a cost to range or speed. Third-class levers are the most common in our bodies, and they place the effort between the fulcrum and the resistance. Our elbow joint with the biceps lifting an object in our hand is a perfect example. The elbow is the fulcrum, the biceps insertion on the forearm is the effort, and the object in the hand is the resistance. These levers are designed for speed and range of motion, allowing us to move our limbs quickly over large distances, even if it requires a greater muscular effort. This elegant system of levers allows our muscles, which can only contract and pull, to create a vast repertoire of pushing, lifting, throwing, and balancing movements. It is another layer of the body's astonishing engineering, optimizing every ounce of muscular effort for maximum functional output. As we conclude our exploration of these fundamental mechanics, it's worth pausing to reflect on the dynamic nature of our muscular system. These contractions, this silent ballet, this orchestral collaboration are not static. They are constantly adapting, constantly responding to the demands we place upon them. Every time we challenge our muscles, whether through a new exercise, a heavier load, or a prolonged effort, we are sending a signal for change. We are inviting them to grow stronger, more enduring, more resilient. This incredible capacity for readaptation is perhaps one of the most inspiring aspects of our muscular anatomy, a testament to the body's ceaseless drive towards improvement and its profound ability to sculpt itself in response to the world it inhabits. In our next quiet contemplation, we will delve deeper into this very idea of adaptation, exploring how our muscles respond to stress, how they grow, and what happens when they are not sufficiently challenged. For now, let us simply hold in our minds the beauty of these contractions, the isometric steadfastness, the concentric triumph, the eccentric grace, and the magnificent symphony of muscles working together, transforming intention into the tangible reality of movement. It is an astonishing universe contained within our own skin, tirelessly performing its silent, powerful dance. And so, we've journeyed through the intricate dance of contraction, the silent symphony of energy, the specialized roles of fibers, the nervous system's masterful conducting, and the ingenious mechanics of lever systems. We've seen muscles as the architects of every deliberate motion, the silent, tireless engines that power our existence. But now, as we turn our gaze towards the deeper currents of their being, we encounter perhaps their most profound and wondrous quality, their astonishing capacity for change. Muscles are not static sculptures, carved once and immutable. Oh no, they are living, breathing, adapting landscapes, constantly resculpting themselves in response to the demands we place upon them, the challenges we present, and even the quiet whispers of time. This inherent plasticity, this magnificent ability to grow, to strengthen, to refine, and yes, sometimes even to recede, is a testament to their profound connection to our lives, our choices, and our very essence. Consider for a moment the gardener who, through years of patient tending, develops forearms like knotted ropes, or the dancer whose calves possess a sculpted resilience that speaks of countless leaps and pirouettes. These are not merely the result of sustained effort, but of a biological dialogue between stress and adaptation. It is here, in this realm of dynamic response, that we truly begin to appreciate the muscle as a living, learning entity. Let us first delve into the phenomenon of muscle growth, or hypertrophy. Picture, if you will, the muscle fiber we've come to know so intimately, that elongated cylindrical cell, packed with its thousands of myofibrils. When we subject this muscle to sufficient stress, particularly through resistance training, we initiate a cascade of events. The mechanical tension itself, the sheer effort of lifting or pushing against resistance sends a powerful signal. This signal is interpreted by the muscle as a call for greater capacity, a need to become stronger, more robust. At a cellular level, this translates into an increase in protein synthesis, a bustling factory floor within the muscle fiber where new contractile proteins, primarily actin and myosin, are meticulously assembled. These new proteins are then integrated into existing myofibrils, making them thicker and stronger, or even leading to the formation of entirely new myofibrils within the fiber. Imagine adding more strands to an already strong cable, or building additional sturdy pillars within a supporting structure. The result is a muscle fiber that increases in diameter, and collectively, a muscle that grows larger and more powerful. But hypertrophy is not a solitary endeavor of the muscle fiber alone. It is a community project. Nestled beneath the basal lamina of each muscle fiber, quiet and unassuming, are specialized stem cells known as satellite cells. In their quiescent state, they are like dormant seeds, waiting for the right conditions to awaken. When muscle fibers are stressed, and even slightly damaged, a natural and necessary part of the training process, these satellite cells are activated. They proliferate, multiplying themselves, and then migrate to the site of need. Here they can fuse with existing muscle fibers, donating their nuclei and cellular machinery, thereby enhancing the fiber's capacity for protein synthesis and repair. Or, under specific conditions, they can even fuse with each other to form entirely new muscle fibers, though this is a less common pathway for typical hypertrophy. Their contribution is profound, essentially expanding the muscle's internal workforce, allowing for greater and more sustained growth. It's a marvel of biological self-improvement, a testament to the body's innate drive for resilience. Yet, just as muscles can grow and strengthen, they also possess the capacity for atrophy, a humbling reminder of their dynamic nature. Imagine a limb immobilized in a cast, or a person confined to bed rest for an extended period. The absence of mechanical tension, the lack of demand, sends a different signal to the muscle, a message that its current capacity is no longer required. The bustling protein factories begin to slow down, and the balance shifts from synthesis to degradation. The muscle begins to dismantle its own components, breaking down contractile proteins and reducing the number and size of its myofibrils. The fibers shrink in diameter, and the muscle as a whole diminishes in size and strength. It is a stark illustration of the principle, use it or lose it, a biological economy where resources are allocated only where they are needed. The speed of atrophy can be quite striking, often occurring much faster than the rate of hypertrophy. Weeks of disuse can undo months of dedicated training, a testament to the muscle's sensitivity to its environment and the constant need for stimulation. This process isn't just about disuse. It can also be triggered by certain diseases, nerve damage, denervation, or the natural process of aging, which we will touch upon shortly. This brings us to the fascinating concept of muscle plasticity, the muscle's ability to adapt its characteristics based on the type of stress it experiences. We've already discussed the different muscle fiber types, the tireless, oxidative type 1 fibers, and the powerful glycolytic type 2 fibers. The remarkable truth is that training can actually influence the characteristics of these fibers, allowing them to optimize for specific tasks. Consider the marathon runner versus the powerlifter. The marathon runner's training, characterized by prolonged lower intensity aerobic activity, stimulates adaptations that enhance endurance. Their type 1 fibers become even more efficient, increasing their mitochondrial density, those tiny cellular powerhouses, boosting their oxidative enzyme activity, and growing a richer network of capillaries to deliver oxygen and nutrients more effectively. Even some type 2 fibers may take on more oxidative characteristics, becoming more fatigue resistant. The muscle, as a whole, becomes a lean, efficient machine, optimized for sustained output. In contrast, the powerlifter's training, with its emphasis on heavy loads and explosive short-duration efforts, drives adaptations for strength and power. This type of training primarily targets type 2 fibers, encouraging their hypertrophy and enhancing their capacity for anaerobic glycolysis. There can even be shifts in fiber type, for instance, from the highly explosive but fatigable type 2X fibers towards the more versatile type 2 fibers, which possess a greater balance of oxidative and glycolytic capabilities, particularly in response to sustained, intense resistance training. The muscle here becomes a powerhouse, optimized for maximal force generation. This specificity of adaptation is truly remarkable, showcasing the muscle's intelligent design and its intricate responsiveness to our chosen activities. But what happens when a muscle is truly challenged, not just by training, but by injury? Here, we uncover another layer of muscle's resilience, its capacity for repair and regeneration. While not as robust as some other tissues in the body, muscles possess a significant ability to heal themselves. When muscle fibers are damaged, say by a tear or strain, an inflammatory response is immediately initiated. This is the body's natural cleanup crew, removing cellular debris and preparing the site for repair. Crucially, those same satellite cells we discussed earlier are once again called into action. They proliferate, migrate to the injured area, and begin the process of rebuilding. They can fuse with the remnants of damaged fibers, helping to repair them, or they can fuse with each other to form new small muscle fibers. Over time, these newly formed or repaired fibers will mature, integrate into the existing muscle architecture, and regain much of their original function. However, the extent of regeneration is not limitless. Severe injuries, especially those involving significant loss of muscle tissue, may lead to the formation of scar tissue, which is fibrous and non-contractile, potentially compromising the muscle's full strength and flexibility. Nevertheless, the very existence of this repair mechanism is a profound testament to the body's enduring drive for wholeness and function. Finally, we must gently acknowledge the quiet, inexorable march of time upon our muscular system. As we age, muscles undergo changes that lead to a progressive decline in mass, strength, and function, a condition known as sarcopenia. This isn't just about becoming less active, though reduced physical activity certainly accelerates the process. Sarcopenia is a complex, multifaceted phenomenon. With age, there's a gradual loss of muscle fibers, particularly the fast-twitch, type 2 fibers, which contribute significantly to power and strength. The remaining fibers may also shrink in size. The nervous system's connection to muscles can also decline, with a reduction in the number of motor neurons and a decrease in the efficiency of neuromuscular transmission. Hormonal changes, such as a decline in growth hormone and testosterone, also play a role in reducing protein synthesis and promoting protein breakdown. Even the efficiency of satellite cells can diminish, reducing the muscle's capacity for repair and regeneration. Imagine a once bustling village where some houses are slowly abandoned, the roads become less travelled, and the younger generation of builders are not as numerous or energetic. That, in a simplified sense, is the gradual effect of sarcopenia on our muscular landscape. The consequence is not just a loss of physical prowess, but a reduced ability to perform daily tasks, an increased risk of falls, and a general decline in quality of life. However, and this is a crucial point of hope and empowerment, sarcopenia is not an inevitable or entirely uncontrollable fate. The same principles of plasticity that allow muscles to grow and adapt earlier in life remain, to a significant extent, throughout our lifespan. Regular resistance training, even in advanced age, has been shown to be incredibly effective in mitigating muscle loss, maintaining strength, and even promoting hypertrophy. Adequate protein intake and overall nutrition also play a vital role. It is a powerful reminder that our bodies are constantly listening, constantly responding, and that our choices continue to shape our physical reality. So, as we conclude this exploration of muscle adaptation, we are left with an even deeper appreciation for these silent architects of our lives. They are not merely bundles of tissue that pull on bones. They are dynamic, intelligent systems, constantly engaging in a delicate dance of synthesis and degradation, growth and recession, repair and regeneration. They are a living record of our efforts, our challenges, and our resilience. They remind us that the human body is a masterpiece of engineering, yes, but also a profound testament to the power of continuous adaptation and the enduring capacity for change. In our next quiet contemplation, we will shift our focus from the skeletal muscles, the voluntary movers we've explored, to the other two remarkable members of the muscular family, the cardiac muscle of the heart and the smooth muscle that orchestrates our internal world. Prepare to be amazed by their unique structures, their involuntary rhythms, and their vital, often unseen contributions to the symphony of life within us. Until then, I encourage you to carry this newfound appreciation for your own muscular landscape, feeling the profound wonder of its ceaseless adaptation with every subtle movement you make. Our journey thus far has largely orbited the realm of the voluntary. These skeletal muscles, those magnificent architects of conscious movement, responding to our every command, shaping our interaction with the external world. We've marveled at their intricate structure, their sophisticated energy systems, their precise neural control, and their astonishing capacity for adaptation and repair. They are the muscles we feel working, the ones we intentionally engage to lift, to run, to embrace. But beneath this visible tapestry of deliberate action lies an equally astonishing, and perhaps even more vital, universe of involuntary muscle. These are the quiet orchestrators, the tireless engines that keep the symphony of life playing, without a single conscious thought from us. Today, we turn our gaze inward, beyond the reach of our conscious will, to explore the unique structures, the intrinsic rhythms, and the indispensable contributions of cardiac muscle and smooth muscle. These are the hidden architects, the silent guardians, ensuring our very survival, moment by moment. Consider for a moment the profound trust we place in these muscles. We don't command our heart to beat, nor do we consciously direct the rhythmic contractions that move food through our digestive tract, or regulate the flow of blood through our vessels. These are processes unfolding in the background, a testament to the body's inherent wisdom and self-governance. It is here, in this realm of the involuntary, that we truly begin to appreciate the full breadth of muscular mastery. Let us first approach the most celebrated of these involuntary marvels, the cardiac muscle. Picture, if you will, the heart, a tireless pump, a fist-sized organ that will beat, on average, over 2.5 billion times in a lifetime, never resting, never faltering. Its rhythm is the very pulse of our existence. Structurally, cardiac muscle shares a superficial resemblance with skeletal muscle, in that it too is striated. Those distinct bands of light and dark visible under the microscope tell us that the myofibrils within are arranged into sarcomas just like their voluntary cousins. The sliding filament mechanism, the elegant dance of actin and myosin, remains the fundamental engine of contraction. Yet the similarities quickly diverge. Cardiac muscle cells, or cardiomyocytes, are typically shorter and branched, forming an intricate interconnected network. Imagine an endless interwoven web, where each fibre is linked to its neighbours, not just end to end, but side to side, creating a functional syncytium, a single, coordinated unit. The secret to this remarkable synchronicity lies in specialised junctions called intercalated discs. These are not mere points of contact, they are sophisticated communication hubs. Within these discs we find two crucial components: desmosomes as gap junctions. Desmosomes act like powerful rivets, firmly binding the cells together, ensuring that the immense mechanical stress of constant pumping doesn't tear the tissue apart. They provide the structural integrity, the physical glue, but it is the gap junctions that truly orchestrate the heart's rhythm. These tiny protein channels create direct cytoplasmic connections between adjacent cells, allowing ions to flow freely from one cell to the next. This means that an electrical signal, an action potential, generated in one cell can rapidly spread to all its neighbours, almost instantaneously. It's like a perfectly choreographed wave, sweeping across the entire muscle mass, ensuring a unified, powerful contraction. This brings us to the most defining characteristic of cardiac muscle, its autorhythmicity. Unlike skeletal muscles, which absolutely require a nerve impulse to contract, the heart possesses its own intrinsic pacemaker system. Specialized cardiac cells, primarily found in the sinoatrial SA node, spontaneously depolarize, generating their own action potentials. They are the heart's internal conductor, setting the tempo. This electrical impulse then propagates through the conduction system, the AV node, the bundle of the Purkinje fibers, and out to the vast network of cardiomyocytes via those crucial gap junctions. This intrinsic rhythm is what allows a heart to continue beating even when removed from the body, provided it has oxygen and nutrients. It is a profound, self-sustaining marvel. While the heart sets its own pace, it is not entirely immune to external influence. The autonomic nervous system acts as a modulator, finely tuning the heart rate and contractile force to meet the body's ever-changing demands. The sympathetic nervous system, our fight-or-flight response, releases norepinephrine, accelerating the heart rate and increasing the force of contraction, preparing us for action. Conversely, the parasympathetic nervous system, our rest and digest mode, releases acetylcholine, slowing the heart rate, allowing for conservation of energy. Hormones too play a role. Adrenaline, for instance, mimics sympathetic effects. This exquisite neural and hormonal modulation ensures that the heart always pumps precisely the right amount of blood, adapting to everything from a peaceful sleep to a strenuous sprint. Given its relentless work, cardiac muscle has an insatiable appetite for energy. It is overwhelmingly aerobic, brimming with an extraordinary density of mitochondria, those tiny biological factories specializing in efficient ATP production. This reliance on oxygen makes it incredibly fatigue-resistant, a necessity for an organ that can never truly rest. And speaking of rest, there's another crucial difference. Cardiac muscle has a remarkably long refractory period. This extended period during which the muscle cannot be re-stimulated to contract is absolutely vital. It prevents the heart from entering a state of tetanus, a sustained, fused contraction. Imagine the catastrophe if your heart muscle locked up like a cramped skeletal muscle. The long refractory period ensures that each contraction is followed by a complete relaxation phase, allowing the heart chambers to refill with blood before the next beat. It's a built-in safety mechanism, guaranteeing the rhythmic, efficient pumping action essential for life. Having explored the marvels of the heart, let us now delve into the even more widespread, yet often overlooked, world of smooth muscle. These are the silent architects of our internal environment, orchestrating countless vital processes without fanfare or conscious intervention. You'll find smooth muscle in the walls of nearly every hollow internal organ. The digestive tract, blood vessels, airways, the urinary bladder, the uterus, the iris of your eye, and even attached to your hair follicles, responsible for goosebumps. Their functions are as diverse as their locations, propelling food, regulating blood pressure, controlling airflow, expelling urine, giving birth, and adjusting the pupil size. Structurally, smooth muscle is strikingly different from both skeletal and cardiac muscle. Its most defining characteristic is its name. It is non-striated. Under the microscope, you won't see those organized bands of sarcomeres. Instead, smooth muscle cells are typically spindle-shaped, wider in the middle and tapering at the ends, and usually contain only a single, centrally located nucleus. Their actin and myosin filaments are present, and they still slide past each other to generate force, but they are arranged in a more diffuse, less organized network, anchored to the cell membrane and to dense bodies within the cytoplasm. Imagine a crisscrossing lattice, rather than the neat stacked rows of a sarcomere. This different structural arrangement leads to a different mode of contraction. Smooth muscle contractions are generally much slower and more sustained than those of skeletal muscle. They can maintain a contracted state for long periods with minimal energy expenditure, a property known as latch state. The regulatory proteins are also different. Instead of troponin, calcium binds to a protein called calmodulin, which then activates an enzyme called myosin light chain kinase, MLCK. MLCK phosphorylates myosin heads, enabling them to bind to actin and initiate the power stroke. This pathway allows for fine-tuning and prolonged contraction. Like cardiac muscle, smooth muscle is entirely just involuntary. But its control mechanisms are even more varied. We broadly categorize smooth muscle into two main types based on their electrical and contractile properties: single-unit smooth muscle and or multi-unit smooth muscle. Single-unit smooth muscle is by far the most common type. Here, the cells are extensively connected by gap junctions, forming a functional syncytium, much like cardiac muscle. This means that an action potential generated in one cell can quickly spread to all its neighbors, causing the entire sheet of muscle to contract as a single unit. Think of it like a wave flowing across a vast interconnected fabric. This type of muscle often exhibits pacemaker activity, where certain cells spontaneously depolarize, generating slow, rhythmic waves of contraction and relaxation. The rhythmic peristaltic movements of your digestive tract, propelling food along, are a perfect example of single-unit smooth muscle in action. The uterus during childbirth also exemplifies this powerful coordinated contraction of a single unit. In contrast, multi-unit smooth muscle consists of cells that are largely independent of one another. They have few or no gap junctions. Each cell is innervated individually by the autonomic nervous system, allowing for much finer, more localized control. Imagine a group of individual artists, each receiving their own instructions, rather than a choir singing in unison. This type of smooth muscle is found where delicate and precise control is required, such as in the iris of the eye, controlling pupil diameter, the ciliary body of the eye, focusing the lens, and the piloerector muscles, causing hair to stand on end. The modulation of smooth muscle activity is incredibly complex, responding not only to the autonomic nervous system. Both sympathetic and parasympathetic can be excitatory or inhibitory, depending on the specific organ and receptor type, but also to a vast array of hormones, local chemical factors, like oxygen levels, pH, and carbon dioxide, and even stretch. For instance, a rise in blood pressure stretches the smooth muscle in blood vessel walls, causing it to reflexively contract to resist the stretch and maintain tone. One of the most remarkable properties of smooth muscle is its plasticity, its incredible ability to maintain tone over a wide range of lengths, a phenomenon known as the stress-relaxation response. Imagine your bladder filling with urine. The smooth muscle in its wall stretches considerably, yet it maintains a relatively constant pressure, preventing premature urges and allowing for significant volume changes without a dramatic increase in tension. This adaptability is crucial for organs that undergo large transient volume changes. It can be stretched to twice its resting length and still contract powerfully, a feat impossible for skeletal muscle. So as we draw this chapter to a close, let us briefly reflect on the magnificent diversity of our muscular system. We have seen 3 distinct muscle types, each a masterpiece of biological engineering, each perfectly adapted to its unique role. Skeletal muscle, voluntary, striated, multinucleated, rapid, powerful, but ultimately fatigable, designed for deliberate movement and interaction with the external world. Cardiac muscle, involuntary, striated, typically unenucleated, rhythmic, tireless, and incapable of sustained tetanus, the unwavering pump of life. Smooth muscle, involuntary, non-striated, unenucleated, slow, sustained, highly adaptable, and incredibly diverse in its control mechanisms, the silent, ubiquitous orchestrator of our internal environment. Together, these 3 types form a living, breathing tapestry of motion and stability, a testament to the profound elegance and efficiency of biological design. From the conscious lift of a hand to the unconscious beat of a heart, from the subtle adjustment of blood flow to the powerful contractions of childbirth, our muscles are truly the silent architect of our existence. They invite us to a deeper appreciation, a patient contemplation of the astonishing universe contained within our own bodies. I'd love to know, as you reflect on these involuntary marvels, what new sense of wonder they stir within you. Do they reshape your understanding of your own body's quiet intelligence? Share your thoughts below, and if this exploration has resonated with you, a quiet like or subscribe helps us continue these peaceful explorations of science in our dreamy academy. For now, let us carry this newfound appreciation for the hidden rhythms of life into our day. We've journeyed deep, haven't we? From the grand, sweeping arcs of a biceps contraction to the intricate molecular dance within a single sarcoma, from the conscious command of a skeletal muscle to the silent, tireless rhythm of our heart and the gentle, persistent persuasion of our smooth muscles. We've seen the astonishing universe of our muscular system unfold, layer by delicate layer, a testament to nature's most exquisite engineering. But here, as our exploration continues, we invite you to step back for a moment, to see these individual marvels not as isolated entities, but as threads in a vast interconnected tapestry, a living, breathing web that defines not just our movement, but our very vitality. For the muscular system, in its breathtaking complexity, is far more than simply the engine of motion. It is a metabolic powerhouse, a silent communicator, a profound influencer of our overall health, engaging in a constant, intricate dialogue with every other system within us. Consider for a moment the sheer metabolic demand of our muscles. Even at rest, they are significant consumers of energy, quiet furnaces maintaining our core temperature, constantly poised for action. When we move, even with the most gentle intention, these tiny biological factories spring to life, demanding fuel with an urgency that underscores their central role in our energy economy. They are the body's primary reservoir for glucose, storing it as glycogen, ready to be called upon. This storage capacity is not merely a convenience, it is a critical buffer, a metabolic safeguard that helps regulate blood sugar levels, a profound and often unsung contribution to our metabolic homeostasis. Imagine your muscles as vigilant guardians of your internal balance, constantly adjusting, constantly responding. But the story of muscles' metabolic influence extends far beyond mere energy consumption and storage. It is here that we uncover one of the most astonishing recent discoveries in muscle physiology, the role of myokines. Have you ever considered that your muscles, when they contract, might be speaking to other parts of your body? Like ancient hermits, they were once thought to perform their work in silent isolation. But we now understand that during contraction, muscles release an array of fascinating signaling molecules, these myokines, into the bloodstream. They are, in essence, molecular messengers, a secret language by which your muscles communicate with distant organs. These myokines are nothing short of revolutionary in our understanding of the body. They travel throughout your system, influencing a cascade of physiological processes. Some myokines possess potent anti-inflammatory properties, acting as gentle balms against systemic stress. Others promote the oxidation of fat, subtly guiding your body towards a healthier metabolic profile. Still others enhance insulin sensitivity, improving glucose uptake not just by the muscles themselves but by other tissues, offering a profound protective effect against metabolic imbalances. And perhaps most wondrously, some myokines even cross the blood-brain barrier, reaching the very seat of our consciousness. Here, they are believed to influence cognitive function, mood, and even neurogenesis, the growth of new brain cells. Picture, if you will, your biceps and quadriceps not just moving you through the world, but subtly enhancing your clarity of thought, lifting your spirits, and nurturing the very grey matter that allows you to contemplate these wonders. It's a breathtaking realization, isn't it? That the simple act of moving our bodies is a profound act of self-care, a systemic symphony of internal communication that elevates muscle from a mere machine to a sophisticated endocrine organ. This intricate dialogue extends outward, weaving the muscular system into the very fabric of our being, in harmony with every other vital function. We've touched upon the obvious partnership with the skeletal system, recognizing bones as the elegant levers upon which muscles exert their force, creating the architecture of our movement. And of course, the nervous system remains the master conductor, orchestrating every twitch, every sustained contraction, translating intention into action with astonishing precision. But consider its profound relationship with the cardiovascular system. Every muscle contraction, particularly in our limbs, acts as a gentle pump, assisting the venous return of blood to the heart. This muscle pump is vital, helping to overcome gravity and ensure efficient circulation, a tireless collaboration that prevents stagnation and nurtures every cell. And let us not forget the heart itself, a muscle unique and tireless, standing as the ultimate testament to muscular endurance and rhythmic perfection. The health of our skeletal muscles, nurtured through movement, directly impacts the strength and efficiency of our cardiovascular system, creating a virtuous cycle of well-being. Even our digestive system, a realm typically associated with internal chemistry, relies heavily on muscular action. The rhythmic wave-like contractions of smooth muscle, known as peristalsis, gently propel food through our alimentary canal, a silent, persistent force ensuring nourishment. And when we shiver, a cascade of involuntary muscle contractions generates heat, a primal mechanism that speaks to muscle's fundamental role in thermoregulation, keeping our internal environment stable even in the face of external chill. Given this profound and pervasive influence, it becomes clear that nurturing our muscular system is not merely about achieving physical prowess. It is about cultivating holistic health. It is about engaging in a continuous appreciative dialogue with our own biology. How do we honor this incredible system? Through movement, of course, not necessarily through strenuous, demanding feats, though those have their place, but through consistent, mindful activity. Regular exercise, whether it's a gentle walk, a session of resistance training, or a graceful dance, sends a clear message to our muscles. You are needed, you are valued. This message in turn triggers a cascade of beneficial adaptations, strengthening, improving metabolic function, enhancing myokine release, and reinforcing the intricate neural pathways that govern movement. It's a celebration of our inherent capacity for motion, a daily affirmation of our living, breathing form. Equally vital is nutrition, the fuel for these tiny biological factories. Just as a master craftsman provides the finest materials for their masterpiece, we must provide our muscles with the building blocks they need for repair, growth, and sustained function. Adequate protein for tissue repair, complex carbohydrates for sustained energy, and a spectrum of micronutrients for enzymatic processes all contribute to the optimal functioning of this astonishing system. It's not about strict diets, but about mindful nourishment, understanding that every bite contributes to the health of our internal landscape. And then there is rest and recovery, the quiet moments of rebuilding. It is during these periods of stillness that the most profound adaptations occur. Muscle fibers repair, energy stores are replenished, and the body consolidates the gains made during activity. To deny our muscles adequate rest is to deny them the opportunity to grow stronger, to heal, and to prepare for the next challenge. It is in the gentle pause that true strength is forged. Even in moments of challenge, our muscles display an incredible resilience. We experience fatigue, that familiar signal that our energy reserves are waning, or that metabolic byproducts are accumulating. But fatigue is not a failure. It is a wise protective mechanism, a gentle nudge from our body to pause, to recover. And in the face of minor strains or the wear and tear of daily life, our muscles possess remarkable capacities for repair and regeneration, a testament to their enduring vitality. The activation of satellite cells, the synthesis of new proteins, the careful orchestration of inflammatory responses, these are all part of the muscle's inherent drive to heal and restore itself. As we near the culmination of our journey through the muscular system, we find ourselves standing in awe of its profound elegance, its astonishing efficiency, and its boundless adaptability. From the powerful contractions that allow us to interact with the world to the subtle, ceaseless rhythms that sustain life itself. Muscles are the very essence of our physical existence. They are the sculptors of our form, the engines of our will, and the silent partners in our metabolic and even cognitive health. This exploration, like all true scientific inquiry, is not an endpoint but a doorway. It is an invitation to carry this sense of wonder, this deep appreciation, into your daily life, to feel the gentle pull of a stretch, the satisfying tension of effort, the quiet recovery of rest, and to recognize the intricate, beautiful symphony playing out within you. The muscular system is a masterpiece of biological engineering, a living testament to the power of design, and a constant source of inspiration for all who pause to truly see its marvels. The journey of discovery, like the quiet work of our muscles, is always ongoing, always inviting us to look a little closer, to understand a little deeper,